Hello, we are back to our lecture series on overview and integration of cellular metabolism, and we will start with a new metabolism, that is nucleotide metabolism. All right. So, we will be covering what do we mean by nucleotides. So, we will be briefly covering a nucleotide chemistry. We will be discussing in this class about de novo synthesis of purine. We will be discussing how the step is regulated. We will be discussing purine salvage pathway as well as about purine analogues. All right. So, this purines and pyrimidines, actually when we refer to nucleotide, the constituent molecules are purines and pyrimidines, right. So, what are they? They are actually nitrogen containing heterocyclic compound, very important, right. So, nitrogen containing heterocyclic compound whose rings contain carbon and nitrogen, right. Generally when we are discussing aromatic ring, we generally think of carbon, right. So, carbon, nitrogen both are present, right. The planar structure of purine and pyrimidine facilitates their close association of stacking, which standardizes or stabilizes double stranded DNA. Means what? These purines and pyrimidines are actually leading to formation of compounds, nucleotides, which are polymerizing to form the unit of life, that is DNA, right. They are also referred to as nitrogenous bases. Okay. So, these things whenever you are asking I mean discussing about DNA, purine, pyrimidine, nitrogenous bases, only one thing should come to your mind, that is the question is being asked about nucleotide metabolism. Okay. So, looking at the structure, this is how they look like, right. In this class we will be focusing on purines, right. Upcoming classes we will be discussing purine, pyrimidines, I mean disorders of purines, and then we will be moving on to pyrimidines. So, pyrimidines actually the structure wise they are simpler, they have got one ring, okay, whereas the purines they have actually two ring. There is one hexagonal ring and there is one pentagonal ring. It can be represented, this pentagon may be present in this side, okay, it may be represented on this side as well, right, but the numbering you should be careful. All right, the hexagonal nitrogen, the ring in the six sided ring, the nitrogen starts as the first one and ultimately it ends with the nitrogen in the pentagon. Okay, fine. So, what are the types of purines that you should be knowing? Adenine, guanine, xanthine and hypoxanthine. All right, these are metabolically important purines. Of course, there are multiple other purine as well. For example, theobromine, theophylline and any other naturally occurring proteins that are present in coffee, cocoa, right. We are not discussing entire nucleotide chemistry to start with, but the very basics which will help you to understand the metabolism of nucleotides, right. So, what are the function of nucleotide? Till now you already have come across nucleotides even if you are not knowing it, right. Number one, DNA, RNA, we all know, right. So, provision of energy, right, in the form of ATP, GTP. What I mean, what does energy help? These energy help in muscle contraction, active transport, ion ingredients, multiple things. So, wherever there is an active energy transport, we talk about ATP and GTP, and this A and G are adenine and guanine, those are nothing but nucleotides, right. Again we have come across these names NAD, NADH, FAD, FMN, right. So, nicotinamide adenine dinucleotide phosphate, adenine, right, flavin adenine dinucleotide, flavin mononucleotide, and even coenzyme A also contains adenosine, right. So, very important, you have come across these names multiple times and all of these are having nucleotide as the main players, right. Activated intermediates, you have read about UDP glucose, UDP galactose, UDP glucuronic acid, S-adenosyl methionine. Wherever these names are coming, again nucleotides are playing an important role. Apart from that, they are active phosphate donors in signal transduction, second messengers, right, very important role, and also via adenylation, uridylation, multiple enzymes are activated. So, you have read, even if you are not told about biological function, just simply by recalling many functions you can already give specific examples, right, I am sure about it, right. So, you should be able to recall where what are the areas where a nucleotide is involved in a metabolism, right. Now, looking at the specific unit of a DNA and RNA, unit of DNA is actually a nucleotide. So, what is a nucleotide? A nucleotide is a purine or pyrimidine base. Okay, those are nucleobases, adenine, guanine, those are nitrogenous bases or nucleobases. When they combine with a pentose sugar, okay, the sugar may be ribose or deoxyribose, right. So, a ribose is how you know glucose looks like in hexagon. Ribose is a pentose sugar, it has got 5 carbons, right. And what is deoxyribose? In case of deoxyribose, in the 2' position there is a H. Now, what is 2', 1'? You can see there is a prime symbol. This is to avoid a confusion where the purine base, the ring of the purine base is named as 1, 2, 3, 4, 5 to 9, and the carbon of the pentose sugar are named as 1 dash, 2 dash, 3 dash, right. So, a nucleobase along with a pentose sugar forms nucleoside, right. And when a phosphate group is attached to it, it becomes a nucleotide. So, nitrogenous base plus phosphate pentose sugar, it becomes nucleoside, and a nucleoside plus a phosphate becomes a nucleotide. All right. So, this fundamental concept should be very clear, and here you can see some example of bases, their corresponding nucleosides and their corresponding nucleotide. All right. So, these are the bases of DNA and RNA. You can choose to memorize this chart, because something, I mean anything from here may be combined as an MCQ question when it comes to all except type or even choosing the best answer type of MCQs. For example, in case of DNA, the base is guanine, the nucleoside is deoxyguanosine, and the nucleotide is deoxyguanylate, and then ultimately it can be monophosphate, diphosphate or triphosphate depending on the number of phosphate bonds. So, what is ATP? ATP is definitely a nucleotide. All right. So, once AMP is formed, one phosphate group, it can be phosphorylated to form ADP and then ATP. All right. So, now that we know what does purine and pyrimidine look like, we have a basic idea about the nucleotide chemistry. We shall now discuss how these nucleobases are actually synthesized. Okay. So, our topic of discussion today is regarding purine synthesis. So, purine can be synthesized in 2 ways. I mean this whole double ring structure can be synthesized in 2 ways. Number 1 is known as de novo synthesis. De novo synthesis means you have heard the term de novo synthesis, where one example is during de novo synthesis of fatty acid. All right. There are multiple examples of de novo synthesis. So, de novo synthesis of purine, like all other de novo synthesis, it happens in our body in situ from very basic metabolites. All primary components are assembled to form a new molecule, just like this new car is being assembled from multiple new components. What is these purines have been seen to be formed from other pathway also, that is known as salvage pathway, which is nothing but recycle of existing nucleotides. So, pre-existing nucleotides if are present, they may be circulated in such a way under action of several enzymes, so that same purine is formed. It can be compared to a situation where multiple parts from a junkyard is assembled to form a functional car, right. Both will help you to drive. So, 2 major routes. Number 1, de novo synthesis, main component is activated ribose in the form of PRPP, phosphoribosyl pyrophosphate, we will be discussing, right. After the amino acid, ATP, CO2, all of them, and there are many more, multiple intermediates are there which forms this, core purine molecule, right. Whereas, in case of salvage pathway, we need the base and then we need the PRPP, and that can actually lead to directly formation of the nucleotide. Nucleotide meaning the nitrogenous base, the phosphate group as well as the pentose sugar. All right. So, when we are considered with de novo synthesis, we should always know what are the sources, and this is a very common MCQ or an image based questions, right. So, you can see this is the number, you can take a snapshot of this and you can actually number the carbon of your own, right, on your own. So, this is the first nitrogen, all right, this is the second carbon, and it goes on and on, where this is the 9th, right, and this is the 8th. So, you can do it, right, you can actually easily do it. I told you the hexagon and pentagon can be represented in either sides. Needless to say, what are the sources? The sources are aspartate, carbon dioxide in form of bicarbonate, right, from the body, glycine, formyl tetrahydrofolate, that is formate, and glutamine, right. We already discussed the intermolecule of glycine is actually incorporated in this molecule, right. So, over here we can see the components that are donating. The first nitrogen is formed the amine of aspartate, the second and 8th carbon are from formate in the form of formyl tetrahydrofolic acid, formyl THF. I told you during one carbon metabolism the folate components are getting interconverted into each other, and the N3 and N9 nitrogen are coming from glutamine, C4, C5 and N7, the 7th nitrogen is coming from glycine, and the 6th carbon is also coming from bicarbonate ion. Okay, I am sorry, this would be 2, this is 6. Okay. So, we are now discussing how this steps are happening, all right. You might find it a bit difficult to remember because there are multiple big names that you need to remember in sequence. Okay. The first step or the preparatory step, it is also called step 0. Why? We will discuss. Is formation of PRPP, right. I told you PRPP is nothing but activated ribose 5-phosphate, because ultimately it is ribose 5. If we already have got ribose 5-phosphate, the entire purine nucleus can be loaded on ribose 5-phosphate and then we can get a nucleotide, all right. It is that easy to remember. So, ribose 5-phosphate plus ATP, the enzyme is PRPP synthase or ribose phosphate pyrophosphokinase, same thing, it forms phosphoribosyl pyrophosphate and a molecule of AMP. So, this is the step 0. Remembering structure not important, I am just showing you the structure so that you can conceptually understand the proceedings. So, why we call it a step 0? We call it a step 0 because formation of PRPP is not exclusive in de novo synthesis of purine. What I mean is PRPP is also needed in the pyrimidine nucleotide synthesis, it is also needed in salvage pathway. So, PRPP is not considered a step in the de novo synthesis, therefore, it is actually the starting molecule, right, just like CPS of urea, right. You can correlate the analogy where it was the starting molecule, but it was not directly participate, the enzyme carbonyl phosphate synthase 1 was not directly a part of the urea cycle. Similarly, here PRPP synthesis not included in the purine synthesis pathway, however, it is a preparatory step or step 0. So, what is the first step? The first step is actually combination of glutamine with PRPP to form glutamic acid or glutamate and 5-phosphoribosyl 1-amine or PRA, all right, phosphoribosyl amine, specifically 5-phosphoribosyl 1-amine. So, this is the structure. Over here you see the phosphate group was already present, right, it has already gone out in the form of PPI, this 2-phosphate goes out in form of PPI and the nitrogen of glutamine gets attached, all right. Here this the entire ribose 5-phosphate has been shown, right, and this is the NH2 group that is attached to the ribose 5-phosphate, all right. You know, in any amino acid the amine group can be represented as NH3 plus and COO minus or NH2 and COH. So, it is one and the same. So, this is the first step by formation of PRA, right, and ribose 5-phosphate is first loaded with the amine group, and ultimately the purine ring will be assembled on ribose 5-phosphate, which we already discussed. So, if you just visualize this step, the further steps will be much easier for you to understand. Next we move to the second step. So, now you can see basically the two rings will be formed one after another, right. So, what happens, the next step is actually glycine incorporation, right. So, what happens over here, the product that is formed is glycinamide ribonucleotide, abbreviated as GAR. The enzyme glycinamide ribonucleotide synthetase, because ATP is involved and one glycine molecule is incorporated. So, what is happening, this NH2 was already present to start with, this is the entire glycine molecule that has been incorporated. So, 2 carbon and 1 nitrogen from glycine is already in, right. This is step 2. In the next step, what happens, if you remembered what were the donors, then you can easily remember these steps also, right. So, next what is happening, a formyl group is being introduced. Formyl group means an aldehyde group or CHO. So, formyl transferase or transformylase, glycinamide ribonucleotide transformylase, it forms formyl glycinamide ribonucleotide. Who is the formyl donor? Of course, N5N10 methylene tetrahydrofolate, 1 carbon donor. This is a one molecule from 1 carbon pool, it is donating 1 carbon leading to the formation of FGAR or formyl glycinamide ribonucleotide. Simply one CHO group is attached. This is step 3. Next, what will happen? Who is the next? That is formation of this, there is no keto group in purine, right, the basic nucleotide. So, there is an NH group. So, this glutamine actually comes in, right, and there is an, whenever NH group is being transferred, NH group is also referred as amide group, right. So, the enzyme is amidotransferase, right, and formyl glycinamide ribonucleotide becomes formyl glycinamidine ribonucleotide, which is abbreviated as M. Okay. So, this is the step where the OH group is converted to an NH group. Okay. Basically, keto acid is becoming an amine form, right. Next, what happens? There is a ring closure, right. So, all the 5 components of the pentagon are being attributed. So, what will happen? With the help of a cyclase enzyme, there is a ring closure and FGM is transformed into aminoimidazole ribonucleotide. So, now we have got a pentagon loaded on ribose 5-phosphate. So, we are halfway. So, we need another few steps by which the entire 6 molecule ring can be formed. Okay. So, let us see what happens. A carboxyl group is introduced, okay, by simple, we know carbon dioxide actually, the bicarbonate actually dissociates to form carbon dioxide and water by combining with H plus ion. So, this donor can be found as bicarbonate in multiple textbooks. Basically, a COO is coming in and it is forming 5-amino 4-carboxy aminoimidazole ribonucleotide, which is abbreviated as ACIR, and aminoimidazole ribonucleotide is abbreviated as AIR, okay, ACIR. This is step 6. So, again we have started forming another ring over here, okay. So, next step, what is happening? An aspartic acid is coming in, right. Again ATP is utilized, the enzyme is synthetized, and the previous intermediate, that is 5-amino 4-carboxy aminoimidazole ribonucleotide, is converted to N-succinyl 5-aminoimidazole carboxyamyl ribonucleotide, which is abbreviated as SAI-CAR or SAICAR, right. Now, do you remember in urea cycle where there was a synthetase enzyme where aspartate came in? Argininosuccinate synthetase. If you have recalled it, very good, because what happens in the next step is very similar to that of urea cycle. Whenever aspartic acid is coming in synthetase, the next enzyme is over there was lyase and fumarate was going out. Same thing is happening over here. So, aspartate has donated its nitrogen and now it has left, the remaining part has left via fumarate, and it ultimately converts to 5-aminimidazole 4-carboxyl ribonucleotide. The succinyl group is gone and this is the final product which is abbreviated as AICAR. This is step 9. We are left with what was the next donor? Again a formyl group. So, same formyl donor, formyl transferase or transformylase, N10 formyl tetrahydrofolic acid. It donates a CHO molecule, right, and it forms just formylated AICR or N-formyl aminimidazole 4-carboxamide ribonucleotide. All right. Again, this is a 1 carbon exchange in which there is a distribution of, incorporation of 1 formyl group. Can you tell me what is the next step? If you have paid attention, the next step is basically the ring closure, right. So, all of these things have already been done. So, ultimately finally is the enzyme is known as IMP cyclohydrolase or IMP synthase, and we get the final product at the end of de novo purine synthesis, which is inosine monophosphate. All right, inosine monophosphate. Now, let me tell you, in majority of cases you may not choose to remember the various intermediates. You can conceptually remember. If you are actually writing all the structures down, it is very easy for you to remember, but even then the name of the intermediates might be difficult for you to remember. These are the changes that are happening. These are the donors in the 5th step and the 10th step. There are ring closure, and in the 8th step there is no extra incorporation of any donor because fumarate is moved, right. These are the abbreviated names of the products that are written over here, and if you are finding it difficult, there is a mnemonic by which you can actually remember this: penguins go fishing, flipping, and amazingly swim and float in water, right. So, this is how I chose to remember, right. This is specifically how I created it. You will not find it any textbook, trust me, and you may remember it in any way you like, but let me tell you, the things that we will be discussing next, that is the later part of this video, that is purine salvage pathway, that is actually more important compared to the intermediate products of de novo synthesis pathway. But what you need to remember about de novo synthesis pathway is how it is regulated. Anyway, we are not done with our purines of interest because we needed AMP, GMP because those are the adenine and guanine are the most important purines, right. But we see that they are not directly synthesized. So, the first intermediate is IMP, right, and AMP and GMP are actually formed from IMP. Okay. Anyway, one thing that you need to know is the processes or the enzymes of purine synthesis. There are 10 steps, right, and in prokaryote each reaction has been seen to be catalyzed by a different polypeptide. All right. There is no problem in that. However, in eukaryotes, like many other such multi-enzyme polypeptide, for example, in fatty acid synthesis, we saw that a single polypeptide was coding for multiple enzymes. Similarly, over here, there are basically 3 polypeptides that are catalyzing multi-channel reaction, multi-catalyst reaction, steps 3, 4, 6, steps 7 and 8 and step 9 and 10. That leads to a phenomenon that is known as substrate channeling. So, what is substrate channeling? It is actually the process of direct transfer of an intermediate between active site of one enzyme and another enzyme, so that a sequential reaction can be done in a biosynthetic pathway that will minimize the time required for substrate to go from one enzyme to another, basically, right. The active site can be located in either separate domain in a multifunctional complex or separate subunit, but what is important is these enzymes are a single polypeptide. So, again these numbers can be formulated as an MCQ question. You need to choose the right combination, that which enzymes are actually acting together as a single polypeptide, right. So, just as we discussed, after IMP is formed, it is converted to AMP and GMP. If we look at the conversion of IMP to AMP, this is often referred to as step 11, right, because actually the first 10 steps which are actually controlled by multi subunit channeling reaction are catalyzed by only first 10 reactions. So, what happens? Adenylosuccinate synthetase active phosphate group is involved, it is GTP. It forms adenylosuccinate and ultimately adenylosuccinate lyase. Again common theme, just like purine synthesis, just like urea cycle, whenever aspartate comes in under a succinate in the synthetase enzyme, a fumarate generally goes out if the motive is just to donate one amide moiety. All right. So, aspartate comes in, fumarate goes out, the names are also same, synthetase and lyase, and ultimately we get adenosine monophosphate, okay, or AMP. When we are converting IMP to GMP, first it is acted upon by a dehydrogenase enzyme which forms xanthosine monophosphate, and ultimately by GMP synthase, okay, it is forming GMP, all right, synthetase, all right, because there is an active high energy phosphate group, that is ATP, is being donated. Now, the beauty of it is when we are synthesizing AMP, we are requiring synthesis of adenosine is requiring synthesis of guanine, and here synthesis of guanosine is requiring synthesis of adenine. So, this will come in handy. So, ultimately AMP will be converted to GMP, ADP and ATP. So, basically AMP synthesis needs GTP or ATP synthesis needs GTP, and GTP synthesis will need ATP, because guanosine monophosphate will also be converted to GDP and GTP, all right. This goes to formation of ATP. So, ATP formation needs GTP and GTP formation needs ATP. This thing you should keep in mind. Why? Because it is the regulation of purine synthesis which is very important, and this is the committed step in de novo synthesis, is actually the first step that is catalyzed by amidotransferase, the first reaction where PAR was formed, and it is being actually inhibited by both AMP and GMP. So, whenever it is a kind of feedback inhibition, when there are excess AMP and GMP in circulation, they will inhibit their own formation, all right. Also, there is formation of, as I told you, formation of AMP from IMP requires GTP, and similarly formation of GMP requires ATP. There is a reciprocal control of production. So, if one is low, another will not be produced, and if one is high, then another will be produced, right. So, thus AMP and GMP inhibit their own formation by feedback inhibition, and they also inhibit formation of IMP from HGPRT, right. HGPRT is an enzyme of purine salvage pathway, right. So, this actually regulation will also be applied when we are discussing the purine salvage pathway. Main thing is ATP and GTP are reciprocally controlling each other, and also the first committed step as well as HGPRT enzyme. So, above the level of IMP production, all right. So, above this level, the generally it is independent and synergistic control, and there is forward activation by PRPP. If there is excess PRPP, there will be excess formation. Once PRPP is formed, there will be excess formation of purine and pyrimidine. And however, below the level of IMP, we saw there is reciprocal control, ATP is controlling GTP and GTP is controlling ATP. So, overall the total amount of nucleotide is controlled, and relative amount of GTP and ATP are also controlled. Now, we move on to a relatively more important but less complicated pathway, that is purine salvage pathway. As I told you, one, that is nucleotides are actually available. We do not need to go through 10 steps to synthesize a ring, but ultimately the the bases are available, and the nucleotides are actually getting attached, formed from the bases directly. Who is helping in? Again PRPP. So, this pathway ensures recycling of purines formed by degradation of nucleotides. So, when nucleotides are degraded, actually the phosphates are gone, the sugar is gone, and nucleobases are remaining. So, they can be actually reused. Here also PRPP is the starting material, Therefore, the level of PRPP actually controls both the de novo and the purine salvage pathway, and hence these pathways are closely related. We should note that the de novo synthesis does not happen in all organs, especially in brain. De novo pathway is not operating. Over there, since this purine salvage pathway is very important, becomes very essential, because still the demand for IMP, GMP and AMP can be met, because all of these reactions are reversible and ultimately AMP, GMP, IMP do not need to be synthesized and resynthesized de novo. So, very important, once we are already having some nucleotides via de novo synthesis, they can be easily interchanged among itself to form the corresponding nucleotides. And what are the enzymes? Hypoxanthine is converted to IMP by hypoxanthine guanine phosphoribosyl transferase, HGPRT. This is the enzyme which both IMP and GMP were inhibiting, right. Next, guanine is converted to GMP by again same enzyme, hypoxanthine guanine phosphoribosyl transferase, and adenine can be converted to AMP via the enzyme adenine phosphoribosyl transferase or APRT. So, basically knowing 3 reaction, we know about purine salvage pathway. We know the regulation? Yes, we also know the regulation that both AMP and GMP will inhibit the enzyme HGPRT, right. So, if there is excess AMP and GMP, we do not need purine salvage pathway as well, but if there is a dearth of these compounds, the nucleotides, purine salvage pathway will be in motion. Now, the last part of this discussion are purine analogues, right. How these purine analogues are actually behaving as anti-cancer drugs. Basically, we saw how the purines look like. Minor or simple modification of these purines will lead to a very structurally similar nucleotide that will be initially falsely recognized by the system or the nucleotide synthesizing system, and then these altered purine nucleotides are actually, purine bases are incorporated in the DNA and RNA, and once they are incorporated, the further DNA and RNA propagation, cell division cannot occur, and thus the cell division is arrested, and thus these purine analogues act as anti-cancer drugs. So, specifically we can see 6-mercaptopurine. Whenever we are hearing the word mercapto, it means replaced sulfur replacement, right. So, 6-mercaptopurine inhibits formation of IMP, conversion of IMP to GMP and AMP. So, it is devoiding the cell of all these intermediates. Similarly, cytarabine or cytosine arabinoside, where the ribose is replaced by arabinose, it kills the cells by getting arrested, by arresting the cell, dividing cells in the S phase. Folate antagonist, for example, methotrexate, it is an altered nucleotide inhibitor of dihydrofolate reductase, hampers with 1-carbon metabolism, so that these 1-carbon groups are not available, and we have seen in many synthetic reaction 1-carbon reaction are almost essential. Therefore, all these drugs, azacitidine, again glutamine antagonist, inhibit steps 1 and 4. So, all of them by inhibiting nucleotide synthesis ultimately inhibits synthesis of DNA and RNA, and thus cancer cells who have got an increased turnover cannot be produced, and thus they act as anti-cancer drug. There are many other examples, but I have quoted a few. So, to conclude, we have discussed the nucleotide chemistry, we have discussed the overview of nucleotide chemistry, we have discussed what are the 10 rather 11 steps of de novo synthesis of purine, we have discussed what are the components from which a purine ring is synthesized, we have discussed the purine salvage pathway, we have discussed the regulation of both of them, and we have also discussed the application of purine analogues in medicine. These are my references, and I thank you for your kind attention.